Datri. Datri?
Ma'am, good morning, ma'am.
Good morning. Good morning.
Ma'am, this is Satyashree.
Oh, Satyasri. Our student, no?
Yeah, ma'am, I'm fine, ma'am. How are you, ma'am?
Fine, fine.
Ma'am, shall I start, ma'am?
Okay. Okay, ma.
Yes. Okay.
Sridevi ma'am, can you start your video?
I have started the.
Please on the video mode, ma'am.
I have on.
Yeah, ma'am.
Yes, ma'am.
You're
Now it
not
is visible.
visible? Oh,
Now it is
for you it
visible,
is.
ma'am.
Okay. Okay, then.
Okay, ma'am. Good morning, one and all. Welcome back for the second section of AICTE ISTE sponsored refresher program on role of artificial intelligence in drug discovery and development. Myself, Satyashree, working as Associate Professor in the Department of Pharmaceutical Chemistry in Vivi Institute of Pharmaceutical Sciences, Gudlawalleru, acting as a chairperson for this section. It's my immense pleasure to introduce speaker of this section, Professor A. Sridevi ma'am. Professor A. Sridevi ma'am is an alumni of Kakatiya University, Warangal, Telangana. Ma'am has 25 years of teaching and research experience, and she served as head of the Department of Institute of Pharmaceutical Technology, Sri Padmavati Mahila Viswavidyalaya. Presently, she is serving as in charge head of Physical Science Department. Dr. Sridevi ma'am specialized in phytochemical analysis, screening of natural products for various biological activities. Proving her scientific competence, ma'am has completed 5 research projects with research grant of around rupees 70 lakhs. Currently, one more project is in pipeline. She has been guiding 8 PhD, guided 6 PhD students, supervised 45 PG project students. Ma'am published more than 60 research peer-reviewed publications and contributed 4 chapters in various books from reputed publishers. Ma'am is life member in professional bodies such as IPA, APTI, ISCA, APP, fellow of ICS, etc. As a BOS chairperson, she played a crucial role in implementation of PCA-approved PG curriculum and introduced MOOCs courses in PG program. Dr. Sridevi ma'am is a recipient of AICTE CAYT Award, UGC Research Award, and State Best NSC Program Officer Award. She is a member in several prestigious bodies, committees like Member Fact Finding Committee for Acharya Nagarjuna University, Selection Committee Member Sri Krishnadevaraya University, IAEC Member Secretary of Sri Padmavati Mahila Viswavidyalayam, IAEC member for several institutions, CPCSA main nominee for Kuppam University, PES Institute, Narayana College, Nellore, and National Assessment and Accreditation Council NAC member 2. She presented more than 60 papers in various national and international seminars, conferences in our country and USA and UK. She is also an editorial board for many peer-reviewed journals. With this brief profile, I welcome you, ma'am, for delivery a talk on computational screening and evidence-based support for in vitro antioxidant activity, Jasminum auriculatum. Welcome you, ma'am.
Thank you, Satyasri, for your nice introduction. Good morning, sir.
Good morning,
Lakshmana,
madam.
sir.
Good morning, madam. How are you, madam?
Fine, sir. How are you, sir?
Fine, madam. Thank you. Thank you for your presence.
Thank you, sir. Thank you for your invitation first.
With
Shall
pleasure.
I start, sir?
Sure, madam.
Yes, ma'am.
Sure, madam.
May I able to present my screen, Satyasri?
No, ma'am. No, ma'am. Please share the screen, ma'am.
Okay. I will.
Madam, after your mic, you have a arrow symbol there.
Open
Yes,
share,
yes,
ma'am.
yes.
That is the share screen symbol. Use that. Afterwards, you have Windows and desktop and Windows is there.
Is it visible?
Yes,
Yes,
ma'am.
ma'am.
Can
Yes,
you
Is
put
ma'am.
it
it
in
Please open
presentation
the
mode?
our.
Is it in presentation mode?
Yes, ma'am. Yes, ma'am.
Shall I proceed now?
Yes, ma'am. You can proceed, ma'am.
Okay. Thank you. So, good morning, my dear participants. So, before going to my topic, that is computational approach and evidence-based support for the in vitro antioxidant activity of Gcms Analysis. First of all, we should know that what is a computational approach. I'm not going to the basics of this computational approach because since 3 days or 4 days onwards, we are going to listening the lectures related to these computational approaches. Now just I will touch only one point why it has been important nowadays. So although this computational approach is prevailing 20 years back, but since 2 to 3 years, it has gained the very, very important, especially in the pandemic situation. As you know that this conventional drug discovery is a time consuming process and costly process. But now whole world is suffering with the COVID-19. So detection or the discovery of the drug by using the conventional methods is very, very difficult within within a short span of time. So the scientists uh, were think alternatively, and they also come up with an idea of drug repurposing. So although the drugs are prevailing and those drugs are used for some other purpose, but they are also invented with the help of this computational approach and a small in vitro and in silico techniques. They identified that they too also having the, apart from that prevailing use, they are also showing the other uses. So what happens is so many drugs are available now in the market. So they tried with the help of this computational approach to treat the COVID-19, and they come up with a very potent drugs also. And nowadays we are using those drugs which are identified based upon the computational approach, then followed by the clinical studies which reduces the time as well as the money, and also they are using successively to treat this COVID-19. By that way, we are reducing the mortality rate. So this computational approach has gained a very, very important in this recent years. So why most of them they are applied for the engineering and as well as for the synthetic chemistry. So why we are not applying for this natural chemistry? So we are applying for the natural chemistry, but that is not that much extent, not that much extent. So with that intention, our natural chemists are also concentrating or taking the help of the computational approach to prove the pharmacological activities of those medicinal plants which are claimed. So with that intention, in our laboratory also, we are performed a study that is nothing but an evidence-based support for the in vitro antioxidant activity of Jasmium auriculatum. Okay. So the next one is, excuse me for 1 minute. Okay. So in my talk, I would like to touch all these points. That is introduction, aim and objectives, plan of work, materials and methods, results, and lastly the conclusion. So before going to the in vitro antioxidant activity, first we should know that what is a free radical. So all of you know that Shit. Excuse me. So the free radical, you all know that the free radical is a molecule or a molecular fragment which contains the one or more unpaired electrons in the outermost orbital. And it is very unstable and highly reactive. Because of that reason only, we are facing many problems with the free radical. Okay, then what are the uses? Although the free radical is having its own uses also. So what are those uses? It is very helpful in the combustion. So with the release of the free radicals only the combustion will occur. And atmospheric chemistry in ozone stratosphere, the ozone layer or the ozone will react with the free radicals. So our ozone layer upon photolysis, the ozone layer will be converted into releases the hydroxyl radical, and which will be helpful for us. And the next thing is polymerization. The third one is polymerization. The free radicals will act as a catalyst for the polymerization. The example is polyethylene. And biochemistry also, it will place a very important role. And some of the free radicals may be formed by accidentally or by the phagocytosis, and which may be used in certain other purposes also. And not only this, these free radicals will also play a very important role in the chemical processes in the human physiology. Although these free radicals are having the having its own uses, but they may also be implicated in the pathogenesis of various diseases. That is the very important thing. So what are the various types of the free radicals that are available? So as you know that superoxide is the one of the free radical, and which is the main source of the generation of the hydrogen peroxide and the hydroxyl free radical, and it may be neutralized by superoxide dismutase enzyme. And the hydrogen peroxide is the another free radical which involve, as it is not a free radical, but it involves in the production of the many reactive oxygen species. Okay, and it may be neutralized by the peroxidases. And hydroxyl radical, this hydroxyl radical is formed by the reduction of the oxygen molecule in the electron transport chain, and it will damages the DNA, lipids as well as the proteins. And the singlet oxygen, it is produced from the hydrogen peroxide and the oxygen and peroxynitrates. Nitric oxide is a gaseous radical, and which plays a very important role in the integrity of the vascular tone. Then how these free radicals will be formed? These free radicals will be formed at the sources of the free radicals. There are two types of sources. One is the one source is endogenous, and the other source is exogenous. And the exogenous is nothing but the UV radiation and air pollution. Next one is ionization of the radiation. And these are the which led to the formation of free radicals, and these free radicals will damages our DNA and may lead to the lipid peroxidation and the protein damage, which ultimately damages the tissues, and tissue damage ultimately damages the organ. And the same thing, the endogenous. Endogenous are the during the phagocytosis or the inflammation will triggers the formation of a free radical, which may also damages the DNA, proteins and lipids, tissue damage. And mitochondria is considered as energy storage or energy store for the cell, and due to this mitochondrial leakage, it is the heart for the metabolism of the various reactions. During the metabolism of the various component, it may releases the free radicals and it may lead to the same thing. And smoking is the another cause to release the free radicals and which may lead to the damage of our tissues. Then our body itself during the metabolic reactions and phagocytosis, the free radicals will be releases. And these free radicals will be neutralized by the antioxidants which are released from our body. Either antioxidants such as vitamin C or the antioxidant enzymes such as glutathione, the superoxide dismutase and catalase. These enzymes will be neutralizes the free radicals from time to time. But in such cases, in some certain cases, where the deficiency of the antioxidant enzymes, if there is a deficiency in the secretion of the antioxidant enzymes such as glutathione, SOD and catalase, or the overproduction of the free radicals will occur. In such cases, there may not be a neutralization will not occur. So there may not be a balance between this free radical production and antioxidant neutralization. In such cases, what happens? The condition arises. That condition is known as oxidative stress. That condition is known as oxidative stress. And this oxidative stress will causes or damages the various organs of our body. So you just see, it will affect the kidney. It will affect the kidney and it may lead to the chronic kidney disease and nephritis and glomerulonephritis. And when this free radical overproduction causes some defect to the skin also, that is nothing but skin aging, sunburn, psoriasis, like that. Certain diseases may be eczema and all these things will be occur. And it will also affect the heart. It may lead to the angioplasty and cardiac fibrosis, ischemia and myocardial infarction. These are the things that may occur when oxidative stress will occur in the heart. And it is not only restricted to these three organs, it will also affect the joints, which led to the rheumatoid arthritis and affect the lungs, led to the asthma. Okay. Then it in the brain, it causes the trauma, cancer, migraine, neurotoxins will be releases, led to the Parkinson's and Alzheimer's disease, etc., etc. Okay. And in multi-organ, it will affect, which led to the diseases like cancer and diabetes, aging, chronic fatigue, inflammation, and all these will be occur. And it also affect the vessels to cause the endothelial dysfunction and atherosclerosis and all these things. And immune system also affect, and which may lead to the cancer and other chronic inflammatory diseases. And it will not leave the GI tract. It will also affect the GI tract, which led to the endotoxin, liver injury and bloating will causes. And eye may also affected by the oxidative stress, which led to a cataract and retinal degeneration and glaucoma. These are the other diseases that may be caused. So in total, whenever the oxidative stress will occur, it will affect each and every part of our body. So we should be very cautious. Okay. Then the antioxidant. Antioxidants are the molecules which are capable to inhibiting the oxidation of the other molecule, because the oxidation will causes the release of the free radicals. So if there is no oxidation, there is no free radicals. And so it will stop the chain reaction which led to the formation of free radicals. Okay. So antioxidants are very, very important. There are various types of an antioxidants based upon their mechanism of action and other things. So, the free radicals will lead to cause the various diseases to the human thing, and the antioxidants are inhibiting these free radicals. Then our scientists were developed the therapies which are based upon both antioxidant and antioxidant enzymes, and they prevented the diseases, and also sometimes they also help to cure the or to treat the diseases. And the antioxidants have a strong potential to be used as a most conventional treatment of a diseases such as inflammatory disease, neurodegenerative diseases, and cancer and diabetic diseases. So the diseases where the free radicals are the main cause or the reason for the occurring of that disease is because of free radical, and those can be prevented by treating them with an antioxidants. So so with this intention, and there were also reports are there, the medicinal plants which are consisting of these antioxidant principles are showed the very effectively prevented or cured the degenerative diseases. So with degenerative diseases where the free radicals are involved. So with a close scrutiny of the literature review, we are decided to, and the phytochemical studies, ethnobotanical data, we are decided to screen the in vivo and in silico antioxidant activity of Jasmium auriculatum. So this Jasmium auriculatum is one of the very widely available plant which belongs to the family of Oleaceae, and the genus is Jasmium and the species is auriculatum. Regarding the vernacular names, in English it is called as an Indian Juhi, and in Telugu popularly known as Adavi Malle, and Sanskrit it is known as Utika. So this upon scrutinization of the standard books, we observed that there were some claims on this plant. The roots were claimed for the ringworm infections, eye diseases, headache, leprosy, mouth ulcers, renal caliculi, burning maturation, and wound healing. And the flowers are also claimed for the cardiotonic and diuretic, and they may also use to treat the ulcers and urolithiasis, nephrolithiasis, and dermatopathy. So with these claims, and also in certain reports were also there on the biological or pharmacological activity of the various parts of this plant. Leaves of this Jasmium auriculatum were shown the diuretic and in vitro radical scavenging and immunostimulatory activity. And the flowers of this plant was reported for anti-lithiatic activity, and aerial pods were reported for antibacterial activity. And recently, one report, 2019, the flowers were also reported for urolithiatic activity of this plant. And certain reports were also there on the phytochemical studies of the various parts of this plant. So the leaves are consisting of terpenoids such as lupiol, limonene, and phytol. And aerial parts were reported for the presence of phenolic compounds such as 4-penta-D-decoxypyrulic acid. And recent reports on the flowers were there, and it was reported for the presence of quercetin. Okay, quercetin. So there are only a few reports are there on the pharmacological screening and phytochemical studies on this plant. And both, if you see that there was no report on the pharmacological activity of the roots, and there was no reports on the phytochemical studies on these roots. Based upon this literature, we decided that although the roots are claimed for the various types of the activities, but no reports till date, there was no reports on that. With that intention, what we are done is we are taken the roots of Jasminium auriculatum for the phytochemical studies and the in vitro and in silico antioxidant activity studies. Okay, the objectives of study is to prepare the ethanol extract of Jasminium auriculatum and to conduct the preliminary phytochemical studies. Then we have to conduct the HPTLC, HPLC and GCMS analysis of this ethanol extract. Then to perform the in vitro antioxidant studies of this ethanol extract for Jasminium auriculatum. Then to correlate the in vitro studies with an in silico studies. These are the objectives of the study. So then regarding the materials and methods, we were collected the roots of Jasminium auriculatum from the Thalakona forest of Chittoor district. And after collecting the roots, it was authenticated with a botanist whose name is Dr. Madhava Chetty, botanist of SV University, and a specimen was also deposited in the botany department of SV University. And after that, then it was washed under the running tap, and then it is shade dried, and after drying it was powdered, and after powdering it was subjected for the preparation of ethanol extract by hot extraction method. Here what we are done is we are taken the powder and macerated and added the ethanol to that and macerated it for 48 hours, and after 48 hours then we were reflexed that plant material along with the ethanol for 3 hours, then filtered, and to that mark again we are added the ethanol, macerated and reflexed, and this procedure that we were followed for thrice, and the filtrate was combined and that filtrate was subjected for the concentration under the reduced pressure, and the concentrate which obtained after the vacuum distillation was stored in the desiccator until it was used. Then after that we were subjected them for the phytochemical studies such as presence of flavonoids, terpenoids, steroids, or we are conducted various types of the chemical tests. Then we were given our sample for the analysis of HPTLC, HPLC and GCMS. And here in the HPTLC, the fingerprint analysis was performed, and here the ethanolic extract was taken, and the extract was dissolved in the ethanol, and the mobile phase used was the chloroform methanol, and ethanol extract of Jasminium auriculatum was 2 microliters. Spot was done, and it was for the development it was placed in that pin trough chamber which is having the measurements of 20 into 10 centimeters, and after developing it was dried. It was dried in an oven at 60 degrees centigrade, and after drying it was scanned at 366 nanometers using the Tamax scanner and WinCast software, and the retention factor of each compound was separated and a plate, and the percentage area of each band was recorded. Then next followed the HPLC analysis, and here in the qualitative analysis, here what we are done is in after preliminary phytochemical studies, it was conducted, it was confirmed that presence of a flavonoid. So then what we are done is we are taken the routine. We would like to know that whether the routine is present in this HPLC or not. So then we are conducted the TLC for the flavonoids in a flavonoid standard mobile phase, that is nothing but ethyl acetate, formic acid and water, and the routine, and we compared that. In that we observed that the routine was given a retention factor which is equivalent to the routine was observed. So by that way we confirmed that the extract is consisting of a routine. So further we would like to confirm that. For that we were given for HPLC studies, and here in this qualitative tests were performed for this HPLC, and the solvent system at the mobile phase used is the methanol and water, and the injected volume is 20 microliters, and the column that was used here is C18 Pyramex column that was used. The flow rate was fixed as 1 ml per minute, and it was detected at 254 nanometers. And further identification of the compounds, we were given the sample for the GCMS analysis. And the instrument here that they were used is Claris 680 GC. And the column is the fused silica column. And the carrier gas is the helium. And the flow rate is 1 ml per minute. And injection volume here is 1 microliter of an extract. And the oven temperature, initially it is 60 degrees, and that was maintained for the 2 minutes. Then every 1 minute they are increased the 10 degrees until it reaches the 300 degrees centigrade. And at that 300 degrees centigrade, they kept for 6 minutes. And electron impact and the spectrums of the compounds which are obtained in this were compared with the database that is stored in the NIST library. Then that is regarding the phytochemical studies. Then next coming to the in vitro antioxidant activity, and that ethanol extract was also performed for the not only for the phytochemical studies, but it is also subjected for the in vitro antioxidant activity. And here we were taken the 3 in vitro antioxidant models. One is the DPPH antioxidant activity, and this method was done by Suri et al. And the other in vitro antioxidant model that we were taken is ABTS, radical scavenging activity, and the method that we were followed is Re et al. That is reported in 1999. And the third and the last in vitro antioxidant model that we were followed here is the nitric oxide scavenging activity. This here the method that we were followed is Srijan and Rao. I think all these methods that you are very familiar because all are very, very famous. So due to that reason, I am not touching how we were performed these methods, but the concentrations that they were used for this screening of in vitro antioxidant activities, the 5 concentrations that we were chosen, namely the 1 mg, 2 mg, 3 mg, 4 mg, and the 5 mg. And the ascorbic acid is used as a standard for all 3 in vitro antioxidant models. Then, after performing this in vitro antioxidant activity, so we would like to know that further whether the lab obtained results in the lab is coinciding the results of the computational approach or not. So this is the introduction. So the process, it is a so here the identified phytoconstituents in HPLC and in GCMS analysis were subjected to in silico evaluation of antioxidant activity by performing the molecular docking studies with two protein targets. We are taking the two protein targets. One is the NADPH oxidase, that is the other name is NOX4, and the superoxide dismutase S4D1 using the Schrodinger glide software. So these are the ligands that we were chosen. I will describe it in detail in the results part. So the compounds which were identified, these are the L1 and from L1 to L16, or the L means ligand, L1, L2, L3. Okay. So these are identified until L1 to L16 is identified in the GCMS, and L17 is identified in the HPLC, and the L18 is the standard. Okay. Then before going to the results, we have to know the preparation of the ligand for the docking. This you are already studied in your previous lectures. In the same way, we are also prepared. That is, the 2D structures prepared ligands were downloaded in the SDF format. So there are various formats here for the Schrodinger. You need to upload the format of SDF only from the PubChem online database, and these molecules were then prepared in Schrodinger lick-prep wizard. And in ligand preparation, all possible conformations were taken into the account. So during the preparation, there are various conformations are possible. So they are taken all into the consideration. And the ligands were then subjected to further pre-docking preparation, where the hydrogens were added, followed by the minimization and optimization of the force field. And finally, in working ligand, directory files were created. Like ligand, the protein has to be prepared before going to be docking. The protein structure codes for the NADPH oxidase, NOX4, and the superoxide dismutase were obtained from the PDB bank, that is a protein data bank, which is available in the online. The proteins were prepared using the Schrodinger's protein preparation wizard by removal of crystallographic water molecules and addition of the hydrogen atoms, followed by minimization and optimization using the force field and Schrodinger. These are the usual preparation of the protein. Then the importance of NADPH oxidase. That is, NADPH means nicotinamide adenine dinucleotide oxidases. These are the multi-subunit enzymes that transfer electrons across the cell membranes. Among the numerous endogenous sources of reactive oxygen species, NADPH oxidase is generally accepted as a major project. So one of the reasons to take up this this protein is this will be the major producer, okay, major producer of the oxidase free radicals which implicated in this. So due to that reason, this was taken. And the other one is superoxide dismutase, and its PDB ID is also there. And the superoxide dismutase. This superoxide dismutase is one of the main reactive oxygen species in the cell. And the superoxide dismutases, these dismutases are the group of enzymes which catalyzes the dismutation of superoxide radical to molecular oxygen. Okay. So, and hydrogen peroxide, providing the cellular defense against the reactive oxygen species. So, due to that reason, this was taken. So, we are taken superoxide dismutase is the one of the protein, and the other one is the NOX4, okay, NADPH. So these two, although so many are available, we are taken these two. And after taking these protein, we are selected the protein, we are selected the ligands, and we are prepared the ligands, we are prepared the proteins. Then we have to go for the grid preparation, and after grid preparation, then we can go for the docking. By applying the Mastro search in glide receptor grid was generated by specifying the binding site residues using the site map tool. Upon preparation of the grid for each protein, ligands were locked to each protein using the extra precision mode, and the docked conformers are assessed by employing the glide score. So first we have to generate the grid, okay, and specify the binding sites using the site map tool. And after preparing the grid for each protein, then we can go for the ligand docking by using the extra or XP. Okay, then the docking conformers may be assessed by glide score. The glide score indicates the how efficiently the protein or the ligand is bind with the protein. Next, coming to the results and discussion. So the first thing, we are prepared the ethanol extract of Jasmium auriculatum, and we followed for the preliminary phytochemical studies. And when we are done the test, except for amino acids and alkaloids, remaining all phytoconstituents such as flavonoids, tannins, glycosides, carbohydrates, saponins was given the positive test. And these are the respective tests that we were performed to whether they are present or not. Okay. So except these two phytoconstituents, remaining all are present. Then this is the HPTLC chromatogram of an ethanol extract of roots of Jasmium auriculatum. And this table shows the various peaks. Okay, so total 7 peaks were observed in the HPTLC, and the starting retention factor is 0.03 to 0.73 is the last one, and the percentage area of the first point of the first peak is high, that is around 24.19, then followed by the third peak whose RF value is 0.10, where the percentage area is 23.68. Okay, we are unable to identify what compounds they are, but it is 7 peaks were given, and the first peak, the maximum percentage area is at 24.19. Then we proceeded for the HPLC analysis. Here in the HPLC analysis, our main intention is whether it consisting of the routine or not. Okay. So it was given a peak at 5.1 reason. So that reason is, you just see 5.189. Okay. A peak was observed. That peak was compared with a pure routine in similar conditions and similar mobile phase. Then it was shown the same retention time. By that way, we confirmed that our extract is consisting of one of the phytoconstituent as the routine. Then we are not satisfied with the HPTLC and HPLC analysis that we were obtained, and we would like to further identify the more and more phytoconstituents from the Jasmium ericulatum. For that purpose, we are also subjected for GCMS analysis. So this is GCMS chromatochrome of a Jasmium ericulatum, and total it was given the 16 peaks, and all these 16 peaks spectra, the spectra that were obtained from this GCMS was compared with that of the NIST library, and based upon that NIST library, so they were provided the names of the compounds and the molecular formula, molecular weight, and so on. What is the percentage of that they are present in that extract? Okay. And the retention time was started at 6.245, and it's the name was started as an ethyl hexyl isopropyl, and the retention time and 30.054 with a cyclotrisiloxane hexamethyl. And these two tables were shown the molecular formula, molecular weight, percentage area, percentage peak area of the respective compounds. So after obtaining these compounds, we searched for the biological activity of the identified the compounds in the GCMS with the help of Dr. Duke's database, biological database. Okay. So with the help of that, the Duke's ethnobotanical database, we come up with certain compounds shows the biological activity. This data is collected from the Dr. Duke's ethnomedicinal database. The ligand 3 is beta D erythropianus was found to be endo aesthetic, beta blocker, anti-cancer, nitric oxide inhibitor, and CNS depressant activity. And alpha trichlorosilane, and it was shown the activity of anti-tumor and nephroprotective and nitric oxide scavenging activity. And alpha-1,3 tertiary butyl peroxyalane was shown for anti-tumor and catecholamine, O-methyl transferase and glutathione transferase and activity. And NF indicate not found. We did not found any activity in that database for this compound. Okay. And the L9 is also found to be abortifacient and lipoxygenase inhibitor, alpha reductase inhibitor, and 5-hydroxytryptamine inhibitor activity. And L10 salicylic acid dimethyl bis ester, it was shown the nephroprotective, diuretic, and certain other type. And L12 hexasterol ditrimethylsilane was used. Biological activity is diuretic and antidote. And L13, whose name is 1,3-bis tetrabutyl peroxyl phthalate, and it is in Duke's ethnobotanical database. It is claimed for antidote, anti-tumor, glucosyl, and glutathione transferase inhibitor, tranquilizer, and topoisomerase 2 inhibitor. And the last ligand which observed in the or identified in the GCMS analysis is the cyclotrisiloxane hexamethyl. It is used for a lipoxygenase inhibitor, alpha reductase inhibitor, analeptic, and angiotensin receptor block, and so on. So there are various other activities were reported for this L16. So, so the compounds, then after that, that is the results of an GCMS, HPLC and HPTLC. Then we were followed the in vitro antioxidant activity of this ethanol extract of Jasmium auriculatum. So we were prepared the 5 concentrations and we observed the activity, in vitro antioxidant activity of ethanol extract of Jasmium auriculatum. They found that in each model also, as the concentration of the extract is increasing, the percentage of inhibition or the antioxidant activity is also increased. You just see increased. 1 mg it is 39.2 and 5 mg it is 51.4. Okay. So like that it was increased. And we were calculated the IC50 values also. So IC50 value for this DPPH is 4.26 mg per ml. And for nitric oxide scavenging, it is 1.35. And for ABTS, it is 1.009 mg per ml. So, in vitro antioxidant activity, it was shown very potent in vitro antioxidant activity. The ethanol extract was shown. The used 3 models, in used 3 models, it was shown the very, very potent in vitro antioxidant activity. So then we would like to observe it in the in silico studies. So for that, first we were taking the NOx4 interactions. Okay. So in this NOx4, the ligand 2 was shown the docking score, glide score is 0.7. So it is a good interaction and it was interacted with an amino acids. It was given the where it was interacted. These are the various amines: serine, glycine, tyrosine, phenylalanine, leucine, okay, histidine. This is the number at which place it is present. At that position of this protein with this enzyme, this ligand was binded with an all these are the hydrophobic bonds. So it is a good score. Okay. So the compound L2 was well bound. Then with this is an interaction of an ligand 4 with an NOx4, and it is also shown the G score of -7, 7.774. It is also very good score, and these are the various amino acids that are involved in the interaction with the ligand. Okay. Then this is the ligand 6 and NOx4 interaction. Here it is binded, but when compared to that of the previous ones, that is L2 and L4, it is less. Yes, it is binded, but the interaction is there. But when you compare with the other compounds, the interaction is somewhat less. Okay. And these are the various amino acids of the protein are involved in the hydrophobic interaction with the ligand 6. And ligand 10 is also having the similar G score to that of the ligand 6. And these are the various amino acids which are involved in the interaction. Then this is another ligand which interacted with the NOx4. Here the score is -7.09. It indicates that it is also very efficiently the good interaction between the ligand 12 and the NOx4. And these are the various amino acids which are involved in the interaction with our ligand 12. Then this is the another ligand, that is ligand 14, and these are the amino acids which involved in the interaction with the ligand 4, and here the G score is -5. So you see that ligand 17, this is the routine. Okay, it is routine, and its G score is -11,559. It is a very high score. So it is a very efficiently the interaction between the ligand 17 and NOx4 is notable when compared to that of the all other ligands. And these are the amino acids which are involved in the interaction of the ligand 17. Okay. Then there is another one. The interaction is -6 only. So when compared to this, our routine is better. Then the second protein, that is superoxide dismutase interaction with an ligands. And first the ligand here, the ligand 8 interaction with an superoxide dismutase is -3, and arginine, valine, serine, aspartic acid, and leucine are involved in the, and other amino acids involved in the interaction with the superoxide dismutase. Okay. And here the ligand 14 interaction was shown in this figure with the superoxide dismutase. And here the docking score is -3.794. And these are the various amino acids involved in the interaction with the ligand. And this is this figure represents the interaction of ligand 17. Here again it is a routine with superoxide dismutase, and here the glide score is the docking score is -4.829. These are the other amino acids that are involved in the interaction with our ligand 17. So when compared to that of the NOx4, the interactions with the SOD is somewhat less. The identified compounds. Okay. So then the overall observation is among all the compounds, L17 and the L14, that is trimethyl 4,2-methyl 4-oxo 2-pentyl phenoxysilane showed the best G score with both the protein targets by forming the strong hydrophobic interactions. Okay, and the marked binding ability of the phytoconstituents with an antioxidant target substantiates the in vitro studies and clearly evidences the antioxidant activity of the roots of Jasmine auriculatum. That is, the marked binding ability of the phytoconstituents with an taken antioxidant targets will substantiate the in vitro studies with the in vivo study or in vitro studies with in silico studies, which shows the strong antioxidant activity of our Jasmine auriculatum. Then the current in vitro and in silico evaluation clearly evidences the antioxidant potential of Jasmine auriculatum, and thus it can be revealed that these roots may have the therapeutic potential in amelioration of many disorders with underlying the pathogenesis of free radical production. So in toto, in summary, as the ethanol extract of the Jasminium auriculatum was shown the good in vitro and in silico antioxidant activity. So this extract may also used to test for the diseases where the free radical involvement is there. So it may be used. It may be screened for anti-cancer activity, Alzheimer's disease, Parkinson's disease, diabetes. Okay, nephroprotector, hepatoprotector, neurodegenerative diseases, cancer. So various diseases, you may test for the various diseases also. In future, it may also show very good activity towards the those diseases where the free radicals are involved. So with this, I'll stop my presentation here. And thank you for the organizers for giving me this chance.
Thank you so much, ma'am.
Thank you. Any questions? Anybody?
Dear participants, if you have any queries, please unmute yourself and ask a question. I request participants interact with Sridevi, ma'am.
Thank you, madam. Thank you for your valuable lecture.
Thank you, sir. So actually, so I need some more targets. I'm waiting for some more targets apart from this SOD and NOx.
Exactly,
But
as you told.
because of time constraint and the personal problem of that person, I'm
Exactly,
unable
madam.
to get, sir. So otherwise, I might have been incorporated those targets also.
Exactly.
I'm sorry for that.
No problem. No problem. With pleasure, madam.
Any queries, dear participants? We get positive feedback from participants, ma'am. Excellent presentation, ma'am.
Thank you. Thank you.
Once again, thank you, ma'am, for sharing your knowledge with us.
Thank you. I thank, first of all, I would like to express my heartfelt thanks to Lakshman Rao sir, who has given me a chance to interact with you. Okay. Thank you, sir. Thank you very
Thank you,
much for
madam.
this
With pleasure.
opportunity. Thank you, sir. And also I thank organizers and your faculty members who introduced me very nicely, especially the Satyasri. Thank
Thank you,
you.
ma'am. Thank
Thank
you, ma'am.
you.
Ma'am, 1 minute, ma'am. On behalf of University of Pharmaceutical Sciences, it's my honor to present an e-certificate, ma'am. Please
Thank
accept
you.
it, ma'am.
Oh,
Yes.
thank you. Thank you.
Please accept my certificate, ma'am.
Okay, thank you.
Thank you, ma'am. Once again, thank you so much, ma'am. Dear participant, please fill the feedback and assessment forms which are already given in your chat box, and all of you assemble back at 1:30 p.m. for the next section. Thank you again.
Okay.
Thank you one and all.
Thank you, madam. I will
Thank
call
Thank
you.
you, ma'am.
Sir.
Okay, madam.
Thank you, sir. Thank you very much,
Okay,
sir.
madam.
Shall I
Okay.
remind, sir?
Yes, madam. You can
Yes.
log out.
Thank you, ma'am.
Okay.
Okay, sir. Yeah.
Dear participants, already assessment link and feedback link posted in your chat box. Please go through those links. And the next section commences from 1:30 p.m. Thank you one and all.
Mama.